Good evening all and welcome to SciSoc's Week 3 talk. Today we have the honor of hosting Professor Sir Tom Blundell. Sir Tom is the current Emeritus Professor of Biochemistry here in Cambridge, having been appointed Sir William Dam Professor of Biochemistry in 1996 and Head of School of Biological Sciences in Cambridge in 2003. Sir Tom was the founding CEO of Biotechnology and Biological Sciences Research Council, also known as BPSRC, under UKRI, and was knighted in 1997 for his contributions to science, with his research focusing on structural and computational biology and their applications to drug discovery.
Sir Tom's talk today will discuss the potential of genome sequences to target cancer and other diseases in today's era of precision medicine via uncovering ideal choices of protein targets. In his talk, Sir Tom will elaborate on how structure-guided approaches in both academia and in the industry paved the way for fragment-based techniques in lead discovery for drug development, especially in ASTEX, a company he co-founded in 1999. Such fragment-based techniques were also used to target mycobacterial infections for patients with tuberculosis, cystic fibrosis, and leprosy. Computational approaches via statistical and machine learning methods have also assisted greatly in the understanding of ligand binding and mechanisms of drug resistance. To hear more about the wondrous developments of these amazing, powerful techniques, let us put our hands together for Sir Tom Blundell.
Well, thank you very much for the very kind invitation and for the nice words. And I'm delighted to be here at what I assume is a multidisciplinary audience. And I will tell my story. It's always a temptation to be a little bit historical, but I did start on the physics and chemistry side and gradually wandered across being head of biology and then found myself in the clinical campus. And all of those areas are relevant to what I'm going to talk about. It's a very multidisciplinary activity. So I'm going to talk about 6 decades. So I started in my early 20s and now in my early 80s, working on these areas. I started working with a Nobel laureate, Dorothy Hodgkin, and at the time I was also running the city council in Oxford, and I was a left-wing politician, and Dorothy Hodgkin, who of course was a famous Nobel laureate, was married to a member of the Communist Party. Well, I was left-wing, but I got interested in what she was doing from my politics and ended up moving from physics and chemistry across to being head of biology and now based in the clinical school. So it's been a very interesting story for me, and I'll encourage you all to be multidisciplinary. So I should say thank you very much for inviting me. So I'm going to talk about genomes only very briefly, but genomes of humans as well as genomes for mycobacteria are absolutely essential to the themes of my own research. So in my company I study cancer, and so human genomes are relevant. And then in order not to get into conflict, I work on mycobacterial diseases in the university. And as you know, it's easy to get venture capital money for cancer. It's very difficult for mycobacterial diseases. So the two are quite complementary. And I go therefore from diabetes, where I started working on insulin, and that was over 60 years ago, and then moving across to cancer, and I'll tell you about my company there, and then in the lab increasingly focus on mycobacteria as we got more sequences. I worked a little bit on the SARS-CoV-2 proteome, but I'm not going to talk very much about that this time. Central to all of what I've done over the past 60 years of science have been the ability to look at molecules, and of course what we're seeing here is cryo-EM, but I started my career with the Nobel laureate Dorothy Hodgkin working with X-rays, and we were the first group to work on a protein hormone just after the myoglobin and hemoglobin work in Cambridge, although I was in Oxford then. And but later on, the X-ray machines have got abandoned, and I'll tell you about cryo-EM and how it's really revolutionized how we can see huge molecule complex. And the temptation always before, if you're interested in biology, was to look at one molecule and see how it acts. But biology is about lots of molecules assembled together, as you can see in this assembly with multiple protein in the components. And if we're really going to target these molecules in drug discovery, we need to know about what they really look like and not how these individual components are on their own. We need both, but we need larger complexes. So one revolution has been cryo-EM, and it's changed the way that almost all of us are working. And in many places that I know, X-ray machines were picked up and thrown in the bin, and everybody works on cryo-EM. But that's not true everywhere, but it has been quite a common phenomenon. And then the other revolution is in computing, and artificial intelligence methods have become really quite widely used. Now, I've written a lot of software, and actually Max Prutz came along to me. You remember Max Prutz is the famous Cambridge structural biologist who worked on lots of important proteins, and he came along to me and he said, "Blundell, what are you doing writing software? Just get back in the lab and do proper experimental work." And I'm afraid I ignored his advice, but all my most cited papers, and some of them have really, 30,000 citations and so on, are all in software. And that's because it's very easy to continue doing some experimental work, but to write a new piece of software, if it's working well, is a much more difficult challenge. So I've been using lots of software. It was statistical methods, but the revolution, of course, now is in artificial intelligence and AI methods generally. And I'll say a bit about those. And that's with the objective of understanding how drugs candidates might bind, and using those to target. And as I said, I mainly worked on cancer in the company I founded, and I'll tell you about that. But many areas, if there are just a few patients, so a genetic disease, or if they're patients from developing countries, capitalism really doesn't work very well. So it's up to us as academics quite often, because there's either too few people suffering from the disease to make it profitable for a company, or the people suffering from the disease may be in large numbers, but they may be poor. And I'm afraid if somebody's founded companies, so I've been involved in capitalism, I really go back to my socialist background when I realised that we have to do quite a lot of things in academia which are not obviously going to make profits and you get a large amount of funding. So I talked a little bit about that, and of course one of the major features is the emergence of resistance, and I spent a lot of time on using software to develop ideas about how those mutations might develop. Now, why is that relevant? Of course, it's relevant because I can then think about drugs which are less likely to be affected by mutations, because not all proteins in a living organism, maybe a bacterium or something, can live without them. So that's become very important, understanding all of this. And so overall, I'll talk not about only about mutations and statistical methods, but also using AI, ML methods as well. So these are the revolutionary changes, and this is my background. I've not really stayed in the same place. I started off in Oxford from a working class family and had no idea what I was going to do. I ended up working with Dorothy Hodgkin, the Nobel laureate, and getting a lot of Nature papers. And then everybody said, you better go away, Tom, and do your own thing. So I moved to Sussex University, which was much smaller. And then to my surprise, when I was, 34, I got a professorship and 200 people. So not too many people who are 34 with 200 people in their lab. And that was really quite a surprise for me, but very exciting. And that was at Brooklyn College. And then I got offered a chair in Cambridge, and I have to say I really didn't want to come. I was involved in politics in London, but my wife is from Africa, from the countryside, and she says London's not much like Matabeleland, that's where she comes from. She's a black African, and so I was forced to take her advice, and I came to Cambridge University. And a couple of years ago, they threw me out of the university because they said I'm too old. And I even got a letter saying, if you aren't out of your lab by tomorrow, then we're going to take everything you own. So I left it right to the last minute to see what they would do. And that happened to be the end of the year, 31st of December. Moved everything on the last day. And so I'm now on the clinical campus, enjoying myself there. So this is the background, but the real background is in the history, because I was very much influenced by this lady, Dorothy Hodgkin, and J.D. Bernal. And J.D. Bernal was quite a left-wing scientist, and Dorothy Hodgkin went and worked with him for a short while. She was quite influenced in politics by her husband as well as J.D. Bernal. And then she came back from Cambridge to Oxford, which is where we were when I started. So J.D. Bernal really got crystals, and he was the first person to realize that the reason why they didn't seem to diffract was that people weren't keeping them wet. And so he suggested, so J.D. Bernal is Dorothy Crowfoot Hodgkin's supervisor in the 1930s, and so Dorothy Crowfoot Hodgkin, together with Bernal, designed this way of keeping protein crystals wet, and suddenly they got amazing diffraction patterns like this. And this really changed everything. They wrote a paper to Nature saying now that a crystalline protein is made to give X-ray photographs. So what they're doing here is keeping the crystal wet in a tube, and this tube has to be with glass or something that is transparent and is also not going to interfere with the diffraction. So I was very, very lucky after 30 years of working on insulin. Dorothy Hodgkin got this, and I was a member of the team in 1969, so not quite 60 years ago, that had this beautiful structure of insulin. So insulin actually has zinc atoms in the center, and then it has 3 dimers arranged around these 2 zincs which are in the center. And I was brought up in a family where nobody had done any science. My grandfather and my father were obsessive artists and musicians, and I suddenly realized that there was art in my molecules which really competed with that in church windows and other places where the symmetry in that contributed to the beauty of the environment. So that was a very exciting time, and I realized that we could use crystals for many, many different targets, but we had to crystallize them. But there were alternatives, and that we could also use cryo-EM and get images there. So you see, these are papers which came out of work in the 60s that we published at the end of the 60s or the beginning of the 70s, which gave the first structures of insulin. And that's me enjoying myself, talking to, this is Dorothy Hodgkin, the Nobel laureate, and amazing person. And I could just sit there with my long hair and fairly radical looks, or maybe not very nice looks. I think now I needed a haircut even more than I do now, and had long discussions about politics and science and everything else in life. And that's how I've enjoyed my science. So the group that I found with Dorothy Hodgkin had another feature which I think is absolutely essential, and that's to have an international group. So the group had people from New Zealand, from Australia, and UK, India, and so on. And I just found that fascinating, and it led me to end up traveling around the world as a scientist. I got elected to 20 different national academies in South America, in Africa, in China, in India, and so on. Not in the US, because they realized that I was close to being a Marxist, and I was banned from their societies. But almost every other academy I became a member of, which was very exciting for me. So here are some more members. Vijayan became head of Indian science, and Ted Baker became head of New Zealand science. They all worked on the structure of insulin that I just showed you, and they became very influential. And we also had great times. So this is what influenced me as a scientist. This is a Nobel laureate, Dorothy Hodgkin, and this is Lord Phillips, who became head of all science in the UK. And these are the 2 people in our lab that really had a major impact. And this is Thomas Hodgkin, who is Dorothy Hodgkin's member of the Communist Party husband, and we were sitting on the ground debating politics as well as debating science, and it was all good fun. I have to say. So this is the story, but going on in parallel, I realized that I was living in a very small house, a very old one, and suddenly there was a plan to build a motorway through the centre of Oxford, and I realised that was crazy. It was historically beautiful, and we really didn't want to have the traffic led from here down and then through the centre and out the other way. We should have another route of taking it somewhere else so that it had less impact on an historical vision. If you've been to Oxford and know how beautiful the main street is, it's only beautiful because we stopped the motorway. And so I stood for politics as a left-wing Labour councillor, and to my absolute surprise, and to everybody else's, I proposed that we pedestrianise the centre, or at least control traffic in the centre of Oxford. A lot of people said you're crazy, you're not an experienced politician. Within 2 years I took control of the city of Oxford, and I did exactly what I proposed. I stopped most traffic going right through the central high street. There were areas where you couldn't drive at all to pedestrianise, and I think they've meant that the centre of Oxford has really been retained in a very beautiful way. I often used to come over here to Cambridge, and I was absolutely amazed. There was no political initiatives to do anything about the city, and traffic was tearing through the city. And that happened for a long time after we'd actually pedestrianised a lot of the centre of Oxford. So that was all very exciting, trying to keep the beauty of central Cambridge, and also the areas of Oxford and the areas of the surrounding areas. So I was an Oxford City Councillor. I basically ran the city. I stopped the motorway coming through the centre. I pedestrianised a lot of the city centre. I made the North Oxford area, where there were a lot of beautiful areas, a conservation area. And everybody told me you can't make a conservation area because it has a city beauty. They have to be very old. But I got my idea of having conservation of a beautiful city through, even if it wasn't several centuries old. And many people followed that. And I introduced bus lanes, and so we had a very interesting time. But then an explosion came in my mind of what was I going to do. I've done all these things, but did I want to be a politician? Did I want to be a modern jazz musician, which I was doing late at nights, or did I want to be a scientist? And I asked my mentor, and she said, Tom, I think you better go away and think about it. And somehow I got a scholarship and to travel. And for a year I wandered around the world. And some of you, if you ever want to do this, realize it's only really easy when you haven't got a family to leave. You can just go for a year or so and wander around. So I went through Europe up to the Trans-Siberian, and that's an 8-day travel, or it used to be on the train, 7 or 8 days. And I got then to the border on the other side of Asia, and I wondered how I was going to get to Japan. And suddenly a beautiful young Japanese lady appeared with a very old Japanese man, and I wondered what on earth is happening. And it then occurred to me what was happening was that the Japanese had been invited back to the scene of the battle between the Japanese and the Russians, which was held here, as the Russians were trying to make friends with the Japanese. So they paid for a plane with this very beautiful young lady, and they paid and brought them over to see the scene of the battle where many of their comrades were killed. And this is the Trans-Siberian. I went across there to Khabarovsk and got stuck, and I then eventually got this plane going with the old man back to see where his comrades were killed, going back to Japan. So I then went to Tokyo, and I lived in Tokyo. So Nihongo Benko Shimashita. In Japanese here, but my Japanese was better than it is now. I lived in Japan for some time, and that's a great experience which you can do when you're young. And then I travelled around and lived a bit in Hong Kong. I couldn't get into China at that time, and I enjoyed a friend of mine who had married a Hong Kong Beijing opera person, and then moved around further and came to India, and there I met all the friends of Dorothy Hodgkin who come from India, and got welcomed there. And what did I do there? I discovered the Veena, so I could already play lots of instruments by ear. I had 3 weeks of piano lessons, and then the piano teacher's husband complained that I was trying to tell the piano teacher that I could play the piano, but I needed proper training, and I started improvising. And he knocked on the wall and said, "I never want to hear that music in my house again." So I studied all sorts of other music, and I studied the Veena, and this is Indian music, some of you may know. And it's one of the most amazing things you can do as a scientist. You can go around the world visiting your scientific colleagues, and you can listen to different kinds of music, and even you can get them. I bought one of these Veenas and had to have a seat on the plane to get it back, and eventually got it back to England and took lessons. So I went back, and then I wondered what I should do, and Dorothy Hodgkin said, I was in Oxford, maybe you better go somewhere else and concentrate on the science. Not too much politics, she said, not too much music, spend some time on the science. So I went to the University of Sussex, which was, Sussex was the place I was brought up. And I focused, and I first got a Nature paper in 1975 on the structure of glucagon. You know that I've been working around insulin, so insulin puts sugar levels down, glucagon puts them up, and they're very different molecules. And then secondly, I did a lot of work on trying to work out how the insulin worked and which parts of them. So this is the insulin molecule, and this part that I've surrounded here is incredibly flexible, and it was clear that when it's binding the receptor that it moved around. So these are papers I published in Nature in my 3 years time at Sussex. But I also did something that caused lots of upset. I submitted a paper on evolution of proteins because it occurred to me that if I had the structure of a protein, I could then argue which of the mutations were going to change the structure and function, and which were conserved because they were critical to the function. And so this was a paper on Darwinian evolution and neutral mutations. And these are absolutely critical, and both of them got published in Nature, so I was really quite surprised by that. So I had quite a lot of things going on, but in parallel I was influenced by this lady, Eleanor Dodson, who, when we were in Oxford, developed a lot of new methods. And then David Phillips, who became Lord Phillips, he did this first structure of an enzyme, turned up in Oxford, and he solved the structure of the first enzyme, lysozyme. And Louise Johnson was one of his team. And David asked us to write a review, and we ended up writing a book. And that's the book that we wrote. So protein crystallography had been pioneered by Nobel laureates, but nobody had written a book on it. And so that's the book we wrote. And it was really quite useful to me, because I learned lots of things I ignored as I was writing it. So here was a change. Dorothy Hodgkin said I'd better go away from Oxford and go somewhere else. And so I ended up going to Sussex, as I said. But then in 1976, so what is it, 50 years almost ago, I became a professor in Birkbeck College. And this was in London. It was good for me as a jazz musician to have a jazz club, the best one in the country, very close by. But I also went to operas a lot, and I've always been an opera fan. And I became the successor to J.D. Bernal, who was an absolutely critical person in the development of structural biology. So then I began to think about a paradigm. How did we go from a gene to a protein, and then if we got the protein structure, how did we go to get a drug? And so this was where I began to go. And first, it was obviously a question of defining biological space. And so one example of that was that I looked at the protein renin, and I could for the first time describe the structure, and this is the long active site. And then there are 3 pockets within the active site with lots of hydrogen bonding. So we wrote a paper in Nature, 40 years ago, really proposing how we would use this for drug discovery. And so it was really to target renin and to design medicines and controlling blood pressure and so on. But as I was looking at pepsin and renin, which are homologs, I realized that I've been looking at a molecule which had some amazing properties. It doesn't look like it, but this half here and that half there actually have the same fold. They're rather distorted, but I suddenly realized I've been looking at it for a year, and then it was staring me in the face. So there must have been another molecule like this, symmetrical. And so I started looking for it, but before I found it, I sent this hypothesis that this existed to Nature. And this was the paper we sent, so not quite 50 years ago, but some time ago. And I proposed that this molecule must exist. And I then proposed that if I found it, this I've been making inhibitors for, and so I could quickly convert the inhibitors from this to that. And you might think that that was a bit crazy because they don't look too similar, but they're evolutionarily related. And I published the paper in 1978, but it was 4 or 5, 6 years before we realized that that molecule was extremely important, and it actually existed. So what was it? It was the virus in HIV, which was causing AIDS. So what we could show was in HIV, which is a human immunodeficiency virus, you get this AIDS, and HIV codes for very few genes, but there was a critical one here, which was the proteinase. Now, you might think that finding a proteinase here is not what I'm doing if I'm trying to work out what how it's functioning. But I suddenly realized that the proteinase in the genome there was controlling the processing of the massive genes that are made. This virus makes a single long chain and it's all cleaved up. If it's not cleaved up, it's not functional. And so I realized if I got the structure of this, which is what I was suggesting that I found, and I proved I found it, then in fact we could inhibit the HIV polyprotein, so it's made as a single chain in HIV, being cleaved, and if you stop that, then it's not functional. And so this is what we did. This is the structure we found of the HIV proteinase. We predicted it first and then published lots of paper, and our work became absolutely central to the design of drugs for HIV. And that was very exciting. We got new drugs proposed, and then I realized there was resistance causing problems, so we worked on that as well. Now, I got up to 1996, and at that time, Cambridge offered me the chair of biochemistry, and with the idea that I'd take over the whole of biological sciences. So I didn't want to come to Cambridge, but my wife, having been brought up in Africa, said that London's not much like Matabeleland, and we're going to Cambridge. So here I came, and so we then started working. That's my wife, by the way. She's quite a famous scientist, very few black women scientists. She has multiple Nature papers and is now retired. But we, between us and this man, Dima Chagasa, who's a friend who originated in Russia, got structures and papers in Science, Nature, Molecular Cell, and then suddenly realized that the future of drug discovery was not really in X-rays but in cryo-EM. And so we began to focus on cryo-EM. So the amazing thing that happened is that this molecule, which is absolutely central to, it's called DNA PK, it's involved in very important pathways, and a lot of people have been making drugs to this, but they didn't know where they're bound. And I suddenly realized that if I didn't use X-rays, but I used cryo-EM, I could probably get, so not 4.3 angstrom resolution, but higher resolution. And so within about 2 weeks, I suddenly found myself with a very high, compared with this, resolution of 2.8 angstrom. It's not actually high for proteins in general, but it was enough to get the details. I could immediately see where all the drugs were and use that to argue how we could improve the drugs. And so these are the people, Amanda Chaplin and Steve Harbury. Amanda is now quite a famous professor, and Steve has done very well as well. And so this was a paper that we published, and these are the sort of things we began to look at. Huge complexes by cryo-EM, and they weren't possible with X-rays. You could never get this huge molecule to crystallize when it's got 5 or 6, 7 different components. However you look at it, and it becomes quite flexible. But we suddenly did that, and we got, as you can see, papers in Molecular Cell, which was very good, and in Nature again. And then we realized we could use cryo-EM for finding drug molecules, even in cases where the protein was very large. So this is Shikang Yang, who has now gone back to a professorship in China. But we could see suddenly how in this huge molecule that I just showed you, there were drugs already targeting it, but nobody knew how they were. And so just 2 years ago, we published this paper where we used cryo-EM to see where the drugs went for this critical target in cancer and other areas, and defined all the structures so we could see the drugs. And I was able to show that you might be able to merge this one with that one and so on, and get even more efficient drugs as nobody had known. So that went into Nature as well, and all very nice. So then, turned the wrong way. So then what we began to do was to work out various software methods. This is a way of looking at the core of a target protein and describing it, and then using this in drug discovery. So we extended the knowledge also, not only by doing X-ray work or cryo-EM, but writing a lot of software. And these are all pieces of software that I wrote, and as I may have said earlier on, Max Krug's found I was writing software and told me to go back into the lab. I continued to write it, but these programs, like Modeler, it's been overtaken now by machine learning, but it was written, as you can see, about 30 years ago or so. This became internationally used with huge numbers of citations and so on. So this was software that I wrote, ignoring the Nobel Prize winner, and it's been used all over the world, as you can see by the large number of citations. And I think these are all out of date a bit now. But then this is the extending the knowledge. So we could then use these modeling programs to take something like Mycobacterium tuberculosis or other mycobacteria I mentioned, and we could model fairly accurately most of the gene structure. Not everything, but quite a lot. And so these then became the basis for making databases. So I produced a lot of databases, and we replaced the suggested by modeling structures, by experimental structures as they came. But we could model most of the genome of most organisms, say in cystic fibrosis and so on. And that helped drug discovery, although I'm not saying the modeling is anything like the experimental work, but it's better than nothing, much better than nothing. And so this is the software, and we also got involved in leprosy. So this is Sandeep Chaitanya, and Sandeep was head of the International Leprosy Mission, and he was working in India. And I suddenly got a message from India saying that he was coming and was going to work in my lab, and he wanted to have the leprosy mission centered in Cambridge. And I was absolutely surprised, especially as he was in the American leprosy mission, and he'd chosen our lab in Cambridge. And so he turned up, and you know, leprosy is one of these secrets. Nobody, if they get leprosy, tells anybody else. The member of the family is usually isolated, and nobody talks about it, because if you talk about it, you lose all your friends and you're isolated as well. And so it's been really a hidden secret how many people having leprosy, and especially in these countries, in South America, Africa and India. And even people I discovered at the time in India, I thought would most likely know what was happening. Most people didn't realize how widely spread the disease was. And so what we did was to do work on that. We also worked on the SARS-CoV-2 genome. I'm not going to talk about that, I'm running a bit behind time. But we could very quickly get large numbers of structures and understand genes from cancer and also all sorts of other proteins. So this was software that everybody thought was not very useful, but it's quite useful until you've got a structure, but you have to understand what it's going to do. So we can predict mutations. So now there was a paradigm that I developed for going from genes to proteins to making new drugs. So the biological space, we use X-rays and proteins, but we could then use the chemical space. And so with this idea, in 1999, so we're moving closer, 25 years ago, I eventually with this man, this is Harry Jyoti, who's been my students about 10 years before this. He and I went and got money, billions in the end, to found a company to work on cancer using structure-guided methods. And Chris Abel, who sadly died, who's a chemist with us, was helping in that direction. And the idea we had was that if you take very small fragments, you could work with 1,000 fragments, work out where they're bound, and you need to do the structure, and then elaborate those so you could get the fragment-based drug discovery. Start with the fragments. And I started with 3 people in the funding in our labs, biology and chemistry. And then what happened is that we grew the company just from a few people within a year or two to have 80 employees originally. It then got up to 300 and then 1,000 spread around the world. And we use this, we built a lab. And the idea was that we started with the small fragments, found out where they're bound, and then elaborated them using structure-guided methods. So this was in 2013. I got a lot of money by borrowing it. I realized I had to sell it. So you might think this is a lot of money, but it's worth much more than that now. And now I can tell you about private business. I, with 2 colleagues, founded the company. We sold it, 14 years later, in 2013, and I got absolutely nothing from it. Other people I borrowed money from got all the profits. So this is, I'm afraid, private enterprise, but at least we got new drugs on the market. And I got drugs going through clinical trials, and then in 2017 I got approval for, or I should say we got approval for a first breast cancer drug, and then another drug for metastatic urothelial carcinoma, and suddenly the company became hugely valuable as everybody saw that we could make new drugs. So the idea, we build from small fragments, and we then tried to use those methods in academia, but to use it on leprosy, which is just a disease of the developing countries, very poor, cystic fibrosis, that's because there are very few people. So those 2 areas in particular, you don't really get many initiatives coming from private enterprise. Our company, we had founded a company, but that was getting cancer drugs. But we could still also, in academia, look at difficult targets in cancer. And that was what we did. So we did tuberculosis, funded by the Gates Foundation. We became the Center for the American Leprosy Mission, and we became the Center for the Cystic Fibrosis Trust development. So all of those occurred over the past decade. This is what we did with tuberculosis. But this is where tuberculosis is really prevalent, and you can see why it's very difficult to get capitalism to work there, because they're mainly poorer people. And this man arrived, Bill Gates and his wife came and talked to me, and I persuaded them that we could do fragment-based discovery. And so he started putting money into a company, and we were developing things for Africa. And this is why it was so important for me, and that's my wife. She's a very experienced PhD, now retired. These are my two children. One got a first-class degree in Oxford, one got a first-class degree in London School of Economics, and these are my relatives in Africa still, and the chances of their having the same possibility of going to university are much less. So we've been designing new drugs for tuberculosis. I'm not going to go through this, but we've been using the fragment-based discovery that we developed, and you use biophysics to see where the fragments bind by screening them, and then you use X-rays to get the structure of the fragment, and then you elaborate them. And you can see there, this is all the fragments found in an active site. And I think any of you could begin to think about how you would make a drug, say, between these two sets, or this one and that one, and which would be the best to target. So this is called fragment-based discovery, and we've got lots of drugs going on the market. This is a surprise visit. I suddenly got a message from Bill Gates saying he wanted to come and see what we were doing, and this was all relevant. And Chris Abel was the chemist, but he sadly died a few years ago, and that's Chris, who's co-founded the company, and myself, and this is Bill Gates coming up the steps in the university. We made Bill put a lab coat on. He wasn't too pleased, but I couldn't display the rules, and so he then came. You can see him there sitting in the corner asking the young things in our group difficult questions, and I was much relieved that I got these guys. But Bill was very, very impressed and funded us quite a lot. And so what we were able to do was to work not only on tuberculosis, but on this Mycobacterium abscessus, which is also important in drug discovery. And we also began to think about the emergence of resistance. So one of the things I was able to do in our group was to think about how these mutations occur in, say, lung cancer or in Mycobacterium abscessus, and in AIDS and HIV protease. And so the software we've written analyzes all of those. And this is a guy I spent the last few days with. He's a brilliant scientist that's still in the group. It's Aaron Parsons. And this is the basis of one of these statistical softwares that we developed. And what you can see is that my lab has been very much dependent on people from all over the world. I did have a party once, and we decided to work out how many languages we could speak. So one of us spoke to one other person in one language. I cheated a bit by starting in English, and then that person spoke to someone else in another language. And in my group I found we could do 30 different languages, and that's because it was really an example of how international science is so important. And so Douglas Pires is obviously not, he's from South America, and this guy, yes, many of people from around the world, India and so on, have been very important. And so this is, well, this is back in academia, what we can do. These are the questions we're still asking, and we're spending a lot of time in working out how we could use the prediction in software for mutations occurring and blocking and binding. We could choose a site on a protein that was less likely to mutate, and I think that's been a very important thing. So here are some of my colleagues. These are people who run the cryo-EM facility. Dima Chiganso is a Russian that I first met his father in Moscow, I think about 50 years ago, and he eventually ended up in my lab, and we had a marvelous collaboration. So these people are the cryo-EM team, and this is my present team. Well, it's about 2 years old now, well, 4 years according to this, forgotten. And you can see that it really is multidisciplinary. There is an English person here, and not too many others. So we're totally multinational. You can also see that influenced by Dorothy Hodgkin, I tried to recruit as many women as I could to get a balance, and that's quite a challenge. As I think everybody realizes, it's much more difficult for a woman who's having children to stay in science, but we can manage it if the employers understand the challenges. So that's thanks to the team, and this is the people. I got thrown out of the university when I was 80, and these people I took with me to the Papworth Hospital, and I'm now part of the university when nobody's looking, but otherwise I'm in part of the university on the campus that's not just academic. And so these are my colleagues at the present time, and they are a mixture of mathematicians, physicists, engineers, computer scientists, chemists, biochemists, and medics. That's of course more than I got here, but that's been the philosophy. So I think I've been going on too long. I'll leave you with that series of thoughts, and you can see how I'm now on the clinical campus, hiding from the university, having been banned. And they said if you don't get out of the laboratory by the time you're 80, we'll take everything you have. Oh, they're really friendly, and I'm writing a book on race, gender, and age discrimination. And so anybody who contributed to that would like to have their name included. I'll very happily put it in. Thank you very much.
Thank you, Sir Tom, for that absolutely wonderful, brilliant talk. We'll now open the floor up to questions. So if anybody has any questions to ask Sir Tom, please go ahead. Yeah.
Make them easy. I won't have a drink. Yes.
At the back, please.
What are the principles by which you manage to get so many people working together in harmony?
Sorry, what was that? How got so many people working together?
In harmony.
Yeah. Well, I've had no problem doing that. I think the trick is that if you're multidisciplinary, it can be a problem if you have areas which are different disciplines, but it has an advantage because you can put together teams where each person has something rather different to contribute. And to be quite honest, I can't remember over the last 50 years any challenges or problems I've had from putting people together. It's always worked. I guess I learned a lot from Dorothy Hodgkin and a lot from David Phillips, who were both pioneers in working groups. But yeah, the reason I love science is not just that it's a challenge, it's that I have multidisciplinary groups working together and they can come from all over the world. Actually, you look at what's the same as my tea in different countries.
Yeah, thank you, Professor. It was a very inspiring talk. So I was also very impressed by how actively you're engaged in like so many different areas besides science. So I'm just very interested in here. Do you have any suggestion as to how to like balance life, passion, and hobbies, and just like how to avoid just being like not as good as like not too good in any of them and just become, you know, a jack of all trades. So I don't know how to say that saying, but yeah.
The trouble is, you get over age, you get a bit deaf. Can you interpret that? Or shall I don't stand up there?
Yeah, just asking how do you balance between all your different hobbies and
Yeah.
passions and scientific interests as well?
Yeah.
How, you know,
How do I balance between my hobbies? Well, coming from the working class family that I was from, my dad was earning £10 a week when I was at school, and so the rule in the family was that everybody had to work. So every morning I got up very early. I started getting up at 5 o'clock. I go and sort out the newspapers in a relative's news shop, and then I deliver the newspapers for about an hour, and then I go back and have a quick meal and go to school. And I did that for many years at this was a grammar school, so-called. And so then the headmaster discovered that a lot of his team was actually doing paper rounds in the morning before coming to school, and he banned them. And I afterwards went back to him and I said, you're obviously not from a working family. If you have to contribute, you may have to get up early. And I got the top scholarship to Oxford, so I haven't been that bad, but so yes, big challenge. I'm not sure that was the question you're asking, but
I would like to ask about there's like quite a lot of paper talking about ProTax and the tax system.
can you hear us get that? I think I go up the side here as well. It's not working.
Do you want to press the button on it?
Oh, okay. Is it more clear?
Yes.
So there's quite a lot of paper talking about ProTax and the tax system. So instead of inhibit the protein function, they do protein degradation by E3 ligase. I was just curious about how do you think about this kind of small molecules in like cancer or other kinds of disease area?
Yes. Trouble with being 82 is that you tend to be deaf. Say it again if you like.
So yeah, okay. So it's curious about the tax system, degradation system by using E3 ligase and small molecules to degrade the protein. What is your thoughts about it? Like, do you think it will work well in the cancer therapy or all kinds of disease areas?
The small molecules are just a start. Yes. So the idea with, I'm not sure what you're asking, but the philosophy behind the fragment-based method is that for every binding site, which is evolved, so not just random, you have a hotspot, and you have to bind to the hotspot to overcome a lot of the entropic terms. So what happens is with the fragment, you get something that binds to the hotspot, and then you keep that, and then you grow it. But if you get something to bind somewhere else, it won't be as valuable because you won't have displaced so many of the critical waters. So what you're doing when you're doing drug discovery is you're not binding to an empty protein.
Yes.
You're binding to a protein with lots of waters bound. And so the affinity of your drug is going to be affected with how efficiently you can displace the waters. So I'm not sure that answers the question.
Thank you very much.
Okay. You want to give that to somebody else? Okay. Yeah.
Can I ask about money?
Sorry.
Can I ask you anything about money? Money.
Asking about money.
Money.
You know that company before,
Yeah.
Aspect, in 2013 when it was sold, you said something that you didn't get much or you didn't get any.
What happened is that because I worked on insulin, there were a lot of companies that worked on insulin, and then that experience was sort of integrated into large pharma. And so very early on, around 45 years ago, I was recruited by almost every large company in Europe and in US to advise them. And so when I wanted to form a company, I had already had quite a lot of experience. And so then I met people in venture capital, and I realized that I could form a company. And then I had this idea about fragment-based discovery. So you get a fragment bind, and then that gets elaborated, and you have drugs. So I've got 3 drugs on the market selling over $1 billion a year. So the method works. But does it work always? It works if you've got something like cancer where rich people are dying, but I don't know how I'm going to get my drugs for third world development funded. I can get something which has all the features of a useful drug, but I can't fund it through clinical trials. And I think that's the real challenge. I mean, how do we get drugs to the world that's not in Europe or US? It's quite a challenge. I'm not sure that I've answered your question. I probably just posed the questions that I'm trying to answer.
Thank you for trying.
Thanks. So obviously didn't.
I think we have time for one more question. After that, if you have any more questions, you can come down and ask. So, any last questions?
We're looking forward to a drink.
Well, in that case, thank you so much, Professor Blundell, for speaking to us tonight.